Welcome to the deep dive. Today, we're plunging into bacterial toxins. These are some, well, truly fascinating and honestly pretty terrifying weapons microbes use. Our mission today is to really help you, our future medical pros, understand how these tiny troublemakers actually work inside the human body. We've pulled together insights from reliable medical sources to give you those key takeaways.
And what's really great here, I think, is we're not just listing facts. We want to help you build a mental framework, so you really get these mechanisms, especially as you're starting out on your medical journey.
Yeah.
We're aiming for those aha moments.
Absolutely, because we know as a first-year med student, it feels like drinking from a fire hose sometimes, right? So today, think of us as giving you a map, some handy tools to navigate bacterial toxins and make it stick. So to kick things off, a big picture question for you to mull over. When we talk bacterial toxins, there are two main groups. Can you name them? And maybe think about one really key difference between them. We'll definitely come back to that.
Okay, before we jump into those categories, let's quickly define toxigenesis. It's simply the ability of bacteria to make toxins. Simple enough. And yes, you're right, there are two main players, exotoxins and endotoxins. Exotoxins. Think of these as like actively secreted weapons. They're soluble proteins made by living bacteria, and they can travel quite far from the infection site to hit specific targets. Very precise.
Okay. So if exotoxins are maybe like guided missiles specifically aimed,
That's a good way to think about it, yeah.
then endotoxins sound more like maybe a landmine, something that's part of the structure.
Exactly. Endotoxins aren't actively secreted. They are part of the bacterial cell. Specifically, it's Lipopolysaccharide, LPS, which is found in the outer membrane of gram-negative bacteria only. These landmines, as you put it, they get released mainly when the bacteria die. Maybe they get killed by our immune system or antibiotics, and then, boom, widespread damage.
Okay, that makes sense. Maybe a quick refresh on gram-positive versus gram-negative would help solidify that.
Good idea. So remember, gram-positives have that thick peptidoglycan cell wall, but no outer membrane. Gram-negatives, on the other hand, have a thinner peptidoglycan layer and, crucially, that outer membrane, which contains the LPS. So just gram-negatives have LPS, gram-positives don't.
Got it. And that brings us right back to our earlier question. The two types are exotoxins and endotoxins. And the huge difference is where they come from, right? Exotoxins are secreted by living bacteria, the sniper. Endotoxins are part of the cell wall of gram-negatives, the LPS released on death, like that grenade, which explains why even dead gram-negative bacteria can cause so much trouble just by falling apart.
Precisely. So let's dig into that endotoxin, that LPS landmine, a bit more. The actually toxic part of LPS is a component called Lipid A. And what's really critical about endotoxins is how our innate immune system sees them. It recognizes LPS as a massive danger signal. Specific immune cells, like macrophages, have a receptor called TLR4, toll-like receptor 4. When TLR4 spots LPS, it's like hitting a giant panic button for the whole body.
Okay, I like the panic button idea. Or maybe like, imagine your immune system is a smoke alarm. LPS isn't just a puff of smoke, it's like setting a huge bonfire right under the alarm.
Yes, that's a fantastic analogy. The response is immediate and overwhelming. That TLR4 activation triggers a huge release of pro-inflammatory cytokines, things like TNF-alpha, IL-1, IL-6. These are chemical messengers screaming danger everywhere. It also activates other systems like the complement cascade, the coagulation cascade, even nitric oxide release, causing vasodilation. The result, widespread systemic effects. Fever, that's why endotoxins are called pyrogenic. They cause fever, then hypotension, so low blood pressure, edema, or swelling. And weirdly, both clotting and bleeding issues. It's a real storm.
A storm indeed. So for endotoxins, let's anchor that with LPS stands for lethal pyrogenic storm. That seems fitting.
Perfect. And this lethal pyrogenic storm is really the core mechanism behind sepsis. Sepsis is this complex, dangerous, body-wide inflammatory response. It's very often triggered by gram-negative bacteria, like E. coli or Pseudomonas, releasing their LPS. That exaggerated TLR4 response leads to tissue injury, organ failure, and eventually septic shock, where the blood pressure just plummets. Another really stark example is meningococcal disease, caused by Neisseria meningitidis. It's a gram-negative diplococcus. Its endotoxin causes a really rapid, intense inflammation, leading to clots and small blood vessels, that characteristic petechial rash you see on the skin due to necrosis, and it can progress incredibly quickly to septic shock.
Wow. So for us future clinicians, spotting those signs, the fever, maybe that specific kind of rash, the low blood pressure, especially if we suspect a gram-negative bug, it's absolutely critical for acting fast in sepsis or meningitis.
Absolutely critical. Early recognition saves lives.
Okay, so that's the systemic chaos of endotoxins. Let's switch gears now to the sniper, the exotoxins, these are secreted proteins, right?
Yeah.
More targeted.
Exactly. They're designed for more specific, often devastating effects. And we can actually categorize them by how they work. I like to use the acronym MAD toxins. First, M is for membrane-damaging toxins. These are also called type 2 exotoxins. They literally punch holes in cell membranes or disrupt them, causing the cell to lyse to just burst open. Think of, say, pneumolysine from Streptococcus pneumonia. Directly damages lung cells.
Ouch. Okay, M for membrane damaging. What's A?
A is for AB toxins. These are type 3 exotoxins. They're pretty clever. They have two parts. The B subunit is for binding. It finds a specific receptor on the host cell, like a key. Once it docks, it helps the A subunit, the active part, get inside the cell. And once inside, that A subunit goes to work disrupting some essential cellular process. Cholera toxin is a classic AB toxin.
Okay, B binds, A is active. Got it. So MA, what's D?
D is for direct signaling toxins, often called type I exotoxins. These include the superantigens. They don't need to get inside the cell. They work by binding directly to receptors on the outside, like MHC class II molecules on immune cells. This forces a connection between the immune cell and T cells that shouldn't happen, leading to a massive, non-specific activation of T cells. Way too many get activated. That causes a huge cytokine release, similar in effect to the endotoxin storm, but triggered differently. Toxic shock syndrome, caused by Staph aureus or strep pyogenes, is the classic example here.
MAD toxins, membrane damaging, AB, direct signaling. That's a helpful way to group them by mechanism. But do they also target specific places?
Oh, absolutely. They're often named for where they cause trouble, too. You have enterotoxins, entero, for gut targeting the GI tract, causing diarrhea, vomiting. Neurotoxins, obviously targeting nerves or the neuromuscular junction, causing paralysis. Pyrogenic exotoxins, which, like endotoxins, can stimulate cytokine release and cause fever. And tissue-invasive exotoxins. These are often enzymes that break down tissues, like collagenase or hyaluronidase, helping the bacteria spread deeper.
Let's zoom in on those neurotoxins. This is where things get really fascinating, I think. You mentioned botulinum toxin and tetanus toxin.
ONT and penospasmin, yes.
The paralysis duo. It's amazing they affect nerves, but in completely opposite ways. How does that work?
It's all about which signal they block. Let's start with botulism, caused by Clostridium botulinum. Gram-positive rod, anaerobic, forms spores, lives in soil, animal guts. You usually get it from food, especially improperly canned stuff, or sometimes in infants, or through wounds. Now, BONT, the toxin, works at the neuromuscular junction, where nerves tell muscles to contract. It specifically blocks the release of the excitatory neurotransmitter, acetylcholine.
Ah, so it stops the go signal?
Precisely. It puts up a permanent stop sign for muscle contraction signals.
And the result is flaccid paralysis. Muscles go limp, weak. You see symptoms like muscle weakness, maybe blurred vision, drooping eyelids, difficulty swallowing, and it progresses downwards. Can even lead to respiratory failure.
This is the cause of floppy baby syndrome and infant botulism, right?
Exactly. And interestingly, we've harnessed this very effect medically and cosmetically with Botox, tiny controlled doses for specific effects. Diagnosis involves clinical picture, maybe finding the toxin. Treatment is supportive care, ventilation if needed, and crucially antitoxin to neutralize any circulating toxin. Okay, so BONT stops the go signal. What about tetanus on the flip side?
Right, tetanus.
Right.
Caused by Clostridium tetani, another gram-positive anaerobic spore-forming rod found in soil manure. Looks like a tennis racket under the microscope sometimes. Gets in through wounds, often deep puncture wounds. Tetanospasmin, the toxin, travels differently. It goes up the nerves to the central nervous system, the spinal cord, the brainstem. And here's the key difference. It blocks the release of inhibitory neurotransmitters, mainly GABA and glycine.
So instead of blocking the go signal, it blocks the stop signal. It cuts the brakes.
Exactly. You've cut the brakes so the motor neurons are constantly firing. The accelerator is stuck on.
Which leads to spastic paralysis, continuous muscle contractions, rigidity, awful spasms, things like lockjaw trismus, where you can't open your mouth, that horrible grimace, the rhizus sardonicus, and the back arching, episotonos, plus high fever, fast heart rate. Sounds horrific.
It is. Diagnosis is usually clinical. The symptoms are pretty unmistakable. Treatment involves cleaning the wound, antibiotics for the bacteria, antitoxin for the toxin, and importantly, muscle relaxants like benzodiazepines to try and control those severe spasms.
Okay, so let's lock this paralysis duo down. Botulinum stops excitatory signals, flaccid paralysis. Tetanus stops inhibitory signals in cystic paralysis, our anchor. BONT is the brakes off. Flaccid tetanospasmin is the throttle stuck. Spastic really drives home why tetanus vaccination is so vital.
Couldn't agree more. Okay, let's move to the gut now, enterotoxins. And the classic example is cholera, caused by Vibrio cholerae. Gram-negative, comma-shaped bacterium, very motile, lives in water, spread through contaminated water or food, often linked to poor sanitation.
And the effect. It's hard to picture the scale. Like you said before, maybe imagine the small intestine turning into a high-pressure hose just spraying out fluid.
That's disturbingly accurate. The cholera toxin is an AB-type exotoxin. The B subunit binds to intestinal epithelial cells. The A subunit gets inside. Once inside, the A subunit flips a switch, basically. It permanently activates an enzyme called adenylate cyclase. This causes a massive buildup of intracellular cyclic AMP, or KMP, and high KMP tells the cell, specifically through CFTR channels, to pump out chloride ions like crazy.
And water follows salt, or chloride in this case.
Always. Huge amounts of water follow those chloride ions out into the intestinal lumen. Plus, it inhibits sodium absorption. The result is that characteristic massive volume rice water stool diarrhea. Patients can lose, it's staggering, up to 20 liters of fluid a day. 20 liters.
20? That's unbelievable.
Leads to severe dehydration, vomiting, metabolic acidosis because you lose bicarbonate, muscle cramps from electrolyte loss, and ultimately hypovolemic shock. It's incredibly dangerous, especially for children.
Okay, that mechanism. Massive fluid loss. Let's anchor cholera. Chloride out, liquid efflux, rapidly activated. COLERA.
Nice one. And treatment reflects that fluid loss. The absolute cornerstone is oral rehydration therapy, ORT. Getting fluids and electrolytes back in is paramount. Antibiotics like doxycycline can shorten the duration in severe cases, but rehydration saves lives. Of course, prevention is key. Clean water, proper sanitation. That's how you stop cholera outbreaks.
And just to be crystal clear, why is clean water the ultimate prevention? It seems obvious, but...
Because the bacterium is transmitted fecal-orally, primarily through contaminated water. If you ensure water sources are clean and sanitation is good, you break that chain of transmission. Simple as that, but requires infrastructure.
Right. Okay, finally, let's touch on something really aggressive. Necrotizing fasciitis, the so-called flesh-eating bacteria.
Yes, a truly terrifying infection.
It's characterized by incredibly rapid destruction of the fascia, the connective tissue surrounding muscles, and some cutaneous fat.
And it's not just one bug, is it? Several can cause this. That's right. Streptococcus pyogenes, or group A strep, is a major player, often involving its M protein. Clostridium perfringens is another, famous for causing gas gangrene alongside necrotizing fasciitis due to its alpha toxin. And even MRSA, methicillin-resistant Staph aureus, can be involved. Often it starts with some kind of tissue contamination or injury, creating a low-oxygen environment where these bacteria thrive. Clostridium perfringens, for instance, ferments tissue carbohydrates, producing gas, hence gas gangrene. The M protein of strep pyogenes can act as a superantigen, causing massive inflammation and also promoting clotting in small vessels, cutting off blood supply. The alpha toxin from Clostridium also damages blood vessels, leading to ischemia and tissue death. People who are immunocompromised, have diabetes, alcoholism, recent trauma or surgery, or use IV drugs are at higher risk.
And the symptoms, what really stands out?
Well, you'll see fever, swelling, maybe redness, or even a purplish discoloration of the skin due to poor blood flow. The skin might feel numb. But the absolute key clinical sign, the major red flag, is pain out of proportion to the visible signs. It hurts much, much more than it looks like it should. That should scream necrotizing fasciitis.
Pain out of proportion. Got it. Let's anchor that. NF, not fair. Rapidly fading flesh. It captures the speed and severity.
Good one. Diagnosis often involves lab tests, high white count, low sodium, high CRP, but ultimately rapid surgical exploration is often needed to confirm it, and treatment has to be immediate and aggressive. Urgent surgical debridement, meaning cutting away all the dead infected tissue, is crucial. That's combined with broad-spectrum antibiotics. Delay is catastrophic. Time is tissue and time is life.
Wow. Okay, that was quite the journey through the world of bacterial toxins. From the systemic inflammation of endotoxins to the precision strikes of exotoxins causing botulism, tetanus, cholera, and necrotizing fasciitis, these microscopic agents really do pack a punch.
They really do. We saw how LPS from gram-negatives acts like that pyrogenic trigger for sepsis, and how secreted exotoxins have evolved. All these different MAD mechanisms damaging membranes, using AB subunits acting as superantigens to cause everything from paralysis to massive fluid loss to tissue destruction.
Yeah.
Understanding why they cause what they cause, the mechanism, that's so fundamental for diagnosis and treatment down the line.
Definitely. And hopefully those anchors we came up with, LPS, lethal pyrogenic storm, the MAD toxins, breaks off throttle stock for the neurotoxins, cholera, and Evatune, not fair. Hopefully those mental shortcuts will really help things stick.
I hope so too.
I am.
And maybe a final thought to leave you with as you continue learning. Think about what the sheer potency and diversity of these toxins tells us about the evolutionary arms race between microbes and us, their hosts. And maybe consider how can we continue to understand, counter, and maybe even carefully leverage some of these incredibly specific molecular actions in medicine in the future.
That's a really fascinating question to ponder, the microscopic battlefield and how we navigate it. Well, thank you for joining us on this deep dive today. Keep learning, keep asking questions, and we'll see you next time.